Long before the pH meter brought acids and bases into clear numerical focus, chemists and physicians wrestled with the mysteries of sour and bitter substances, fermentation, and bodily fluids. In the late 18th century, the French chemist Antoine Lavoisier laid the groundwork for our understanding of acids by showing their common element, oxygen, while his contemporary, Guyton de Morveau, catalogued dozens of acids and bases in his laboratory manuals. Yet there was no simple way to compare one solution to another except by taste, color changes in rudimentary indicators, or unwieldy titrations. By the turn of the 20th century, indicators like litmus paper could distinguish acids from bases, and more refined dyes such as phenolphthalein or methyl orange gave a crude endpoint for titrations. But these methods were qualitative at best: red or blue, pink or yellow, acidic or alkaline. Danish chemist Søren Sørensen, working at the Carlsberg Laboratory in Copenhagen, sought a more precise measure. In 1909, while studying the behavior of proteins in brewery fermentation, he introduced the concept of pH to denote the negative logarithm of the hydrogen ion concentration. With pH, solutions could be compared quantitatively, pH 2 10 times stronger than pH 3, and so on. His scale from 0 to 14 quickly proved invaluable across chemistry and biology, even if it still relied on indicators and glass electrodes used as rudimentary voltmeters. The real leap came in 1906 when the German chemists Fritz Haber and Zygmunt Klemensiewicz fashioned the first prototype glass electrode. They discovered that a thin membrane of special silicate glass, when immersed in different pH solutions, developed a tiny electric potential proportional to the hydrogen ion concentration difference across the membrane. But the equipment to measure such minute voltages accurately, vacuum tube amplifiers and sensitive galvanometers, didn't yet exist in a practical, portable form. Thus, the glass electrode remained an elegant laboratory curiosity rather than a bench tool. Over the next 2 decades, improvements in glass chemistry produced electrodes more robust and sensitive. Meanwhile, advances in electronics, particularly the development of reliable vacuum tubes, made it possible to amplify and read millivolt signals. Yet solutions still had to carry bulky apparatus, benchtop amplifiers, temperature baths, and delicate reference electrodes. Field measurements were nearly impossible and clinical applications were limited. Everything changed in the early 1930s when Arnold O. Beckman, a young professor at the California Institute of Technology, was approached by his former student and friend, the food scientist S.C. Kelly. Kelly described the difficulty of measuring the acidity of citrus juices during the citrus boom in Southern California. Commercial fruit packing houses needed a simple device to control quality and standardize products, but the existing options, titrations and paper strips, were slow and imprecise. Beckman saw an opportunity to apply electronic amplification to the glass electrode concept. In 1934, after months of experimentation in his Pasadena garage laboratory, Beckman introduced the Model G glass pH meter. It combined a dependable glass electrode and a calomel reference electrode with a vacuum tube amplifier and a moving coil meter calibrated directly in pH units. For the first time, a technician could dip the electrodes into a sample of lemon juice, turn a knob, and read the acidity on a dial within seconds. The instrument was portable enough for factory floors and robust enough for daily use. Within a year, Beckman instruments sold thousands of units, revolutionizing not only the citrus industry but also brewing, dairy, pharmaceuticals, and eventually clinical laboratories. The pH meter quickly became indispensable. In medicine, it enabled reliable blood gas and acid-base balance studies, transforming our understanding of physiology and critical care. In environmental science, researchers used it to monitor river acidity, detect industrial pollution, and study soil chemistry. In metallurgy and electroplating, workers controlled baths to nanometer levels of acidity. And in teaching laboratories worldwide, students learned acid-base chemistry through real-time measurements rather than static color charts. Through the mid-20th century, refinements abounded. Solid-state electronics replaced vacuum tubes, shrinking the meter and reducing warm-up time. Temperature compensation circuits corrected for fluctuations that once introduced errors of whole pH units. Novel electrode materials, combining glass with polymers or crystalline crystals, extended the pH range into highly acidic or alkaline extremes. Miniaturized sensors allowed inline process monitoring in chemical plants. Portable, battery-powered meters put pH measurement into clinicians' pockets and onto oceanographic vessels. In the 1980s and 90s, digital displays and microprocessors transformed the user interface. Automatic calibration routines eased operator burden, while data logging modes recorded pH changes over hours or days. Today's handheld and benchtop instruments can measure pH, ion concentration, conductivity, dissolved oxygen, and even redox potential all in one package. Wireless probes transmit data to cloud-based systems, alerting engineers to pH deviations in remote water treatment plants or fermentation tanks. Yet despite dazzling technological progress, the heart of the pH meter remains the same glass membrane, the same tiny voltage, the same logarithmic relationship discovered by Sørensen and tapped by Haber, Klemensiewicz, and Beckman. In classrooms, hospitals, laboratories, and factories around the world, analysts still calibrate against buffer solutions at pH 4, 7, and 10, dip their electrodes into samples, and read out the result. The pH meter story is one of careful observation, cross-disciplinary insight, and the marriage of chemistry and electronics, a journey that turned the invisible world of hydrogen ions into a chartable, controllable, and comprehensible dimension of modern science.